Hi everyone, in this PowerPoint I am going to walk you through a visual online tool case study that will help us understand cystic fibrosis, and then we'll talk about specifically diagnosis of CF, treatment, and also the molecular pathology of cystic fibrosis. And this will help us in understanding what you'll need to write for your research paper section on molecular pathology in particular. And then we'll finish this PowerPoint up by describing a little bit of some of the alternative approaches to cloning human disease genes, talking about GWAS, genome-wide association studies, and some other approaches. So especially for complex diseases, you'll probably see that their approach to cloning the gene associated with the disease is quite different from the approach used for CF or monogenic diseases. And this last section of the PowerPoint should also be helpful for your assignment that's due on Thursday, April 2nd. Okay, so at the bottom of this slide is the URL where I found this nice case study that talks about cystic fibrosis, and then in particular it's going to help us understand gene therapy and considering whether gene therapy might be appropriate to treat cystic fibrosis. Unfortunately, the case study uses Flash, which works on old Macs and PCs, all PCs. It should work on all PCs, but not on the new iOS. So what I've done here is pasted images from this Flash file into the PowerPoint. If you have Flash on your computer, you can just go to the URL below, and it's an interactive site, and you may find that to be a little bit nicer than just looking at the PowerPoint files. But for those of us, including me, who can't use Flash right now, we can just look at these still images from that file. So first we have an introduction to cystic fibrosis. It's a hereditary disorder. It affects the cells of the lungs, pancreas, and small intestines. And what happens is that abnormal mucus secretions in those tissues cause symptoms such as coughing, poor digestion, and increased vulnerability to infection. Most untreated cystic fibrosis individuals will develop fatal lung infections and not live past their late 20s. And the key term there is untreated cystic fibrosis individuals. I've added the updated life expectancies for CF patients who do get treatment, and you can see that the median predicted survival age, depending on the year of birth, has increased quite a lot. So that is good news for CF patients, although obviously it's still not a full life expectancy. Okay, so if we if in the interactive, you would kind of zoom in to the cells and the tissues and see that the cells that line the airways in the lungs produce that abnormal mucus, and that this is due to a mutation in the CFTR gene on chromosome 7. And we're familiar with chromosome 7 being the location of the gene from that Watkins paper. Most commonly, the mutation in the CFTR gene is a 3 base pair deletion, which is shown here. The mutated CFTR gene has 4,440 base pairs, in contrast to the normal gene that has 4,443. And this mutation was identified in the paper by Rorden et al. In 1989, and I've described that in the 32620 PowerPoint file. So it's important to realize that when there's a mutation in a gene, that's going to affect the protein. So here you can see the normal protein on the right, and then on the left is the protein made from a mutated CFTR gene. And so you can see that the shape and the structure of the protein have changed due to that missing phenylalanine. So just one amino acid is missing, and yet the shape and structure of the protein changes pretty dramatically. And certainly the function of the protein changes. Okay, so we'll come back to function in just a few slides, but first I'm going to talk about diagnosis, because this is one of the areas that you're writing about for your draft of parts 1 through 3. So cystic fibrosis is diagnosed in newborns. Every state in the United States now routinely screens newborns for cystic fibrosis, which is really good news because early diagnosis means treatment can begin very early. One of the things they screen for in the blood samples is high levels of a chemical released by the pancreas called IRT, or immunoreactive trypsinogen. Now, there could be some false positives, some reasons other than cystic fibrosis to have a high IRT level. So they need to perform subsequent tests to confirm a diagnosis of cystic fibrosis. They can do sweat tests and they can also do genetic testing, right? Because we know the exact gene and it's a monogenic disease. You can sequence the CFTR gene and see if there are mutations. In Michigan, the newborn screening program, in case you didn't know about it, it's pretty interesting. It's a public health program. It's required by Michigan law. And the idea is to try to identify babies that have rare but serious disorders. So all babies have to be tested, again, by law. And babies that have particular conditions, but they seem healthy at birth, but they can become sick in a short time, it's really going to help out those babies. Every year in Michigan, more than 250 babies are found to have a disorder that's detected by this newborn screening. And they call it blood spot screening, and I'll show you a picture in just a minute of kind of how that works. You can go to that website at the bottom to see the list of disorders that are screened for in infants in Michigan. Every state has a slightly different list of diseases for which they're screening. Okay, this blood spot screening occurs in this way. They do a heel prick, and you can see the little baby's foot there in this picture, and blood is coming out of the heel, and they literally get these blood spots on a special piece of cardboard, and those are going to be tested for all the different diseases, depending on the state. Diagnosis can also occur via prenatal screening. So for cystic fibrosis, you can get carrier screening done before or during your pregnancy to find out if you're a carrier. If screening shows that you are a carrier, and that would mean you have a mutation in one of the two CFTR genes, your partner could also have screening done. And this really can make a lot of sense because about 1 in 31 people in the United States is a carrier of cystic fibrosis. So if you and your partner are carriers, you can do prenatal testing to see if your baby has CF. And you can do this in 2 different ways, either chorionic villus sampling, which can be done at 12 weeks, and that's assaying the tissue of the placenta, or amniocentesis can be done at 15 to 20 weeks. And that's a diagnostic test where a small amount of amniotic fluid is removed. You can see that long needle inserted into the mother, and then that fluid can be tested to see if there's a genetic abnormality. In terms of treatment for cystic fibrosis, there's not currently a cure. So most of the treatment focuses on easing symptoms. So to prevent lung infections, there are antibiotics, mucus thinning drugs, bronchodilators. There's also chest physical therapy, again, to try to loosen up the mucus that accumulates in the lungs. There's pulmonary rehab, exercise training, breathing strategies. There's also treatments to try to prevent or treat intestinal blockages. And there are also oral pancreatic enzymes for nutrient absorption. More recently, there are newer medications on the market that actually specifically target the CFTR gene. And this is really exciting. You know, when we talk about why do we need to map and clone genes, and we say, well, this is so that we can develop treatments, it's partly to understand, you know, what exactly mechanistically is going wrong in these patients, but it also allows us to target these new drugs, you know, to a specific gene or protein. So in this case, there are now medications that actually help improve the function of the faulty CFTR protein. And they talk about these 3 drugs in this bullet pointed list. I don't know if I can pronounce them, but there's one Elexacaftor, Ivac after, and Tzac after. Okay, and so those 3, it's a combination of 3 different drugs, and it goes by the name of Trikafta. So this is thought to be very exciting and is considered a breakthrough. So I was curious about exactly what are those drugs doing to help improve the function of the mutated CFTR protein. And so here I've just shown you what I learned, that 2 of those drugs, the E drug and the T drug, they bind to different sites on CFTR protein. And together they can help facilitate cellular processing and trafficking to actually get the defective CFTR protein to get to the cell surface. So that is apparently part of the problem in why it's not functioning properly, is it's not even getting to the cell surface. And then the third of those three drugs, the I1, it potentiates the channel open probability. So remember the CFTR encodes an ion channel, a chloride ion channel. So this is actually a drug that can help the gating of that ion channel. And in combination, those three drugs are apparently can result in increased quantity of those ion channels getting to the cell surface and then also increase their function. And in total, this results in increased CFTR activity. And they've been able to measure that by looking at chloride transport and some other assays to measure and show that it's effective. And the bottom line is, in the patients who take this sort of cocktail of three drugs, that their symptoms do improve markedly. So that's exciting for like sort of quality of life and potentially life expectancy. Okay. So the next question is, would gene therapy for cystic fibrosis make sense as a treatment, right? So is this disease a good candidate for genetic or molecular treatment? So we just talked about one way in which it clearly is a good candidate, but on this slide, there are 5 questions that I want you to address in part 3C of your research paper. So that is something that you're going to want to include in your draft that is due on March 31st. The 5 questions that you want to consider are, does the condition result from mutations in one or more genes? So basically, is it genetic? Are the genes known? Like which genes are involved? Number 3, what's known about the biology of this disorder? Number 4 is a really important one. Will adding a normal copy of the gene fix the problem in the affected tissue? And number 5, is it feasible to deliver the gene to cells of the affected tissue? So you would want to see that there are that you would have answers of yes to all 5 questions in order for the disease to be a really good candidate for gene therapy. So those are things to think about for your disease, and if you have any questions about how to think about those, you can ask me. Okay, so what's known about the molecular pathology of CFTR? What's the function of the wild-type protein, and how does the mutant protein result in disease? So basically, in what way are the cells not functioning normally? And are there any other proteins affected? Is there any sort of pathway? So when we come back to our case study, it very nicely shows us first the normal function of the protein. So in normal cells, the CFTR protein functions as a channel and it's moving chloride ions across the cell membranes. And in this view and the next couple slides, the inside part of the cell is shown at the top, and outside the extracellular area is shown at the bottom. Okay, so we see the chloride channel there in sort of yellow color, and there's also a sodium channel in green on the right. So normally, the CFTR chloride channel works with these other channels to set up an ionic gradient, which is going to be a difference in the number and type of ions that are inside versus outside. So we're looking for a balance between the chloride and sodium ions. Normally, the cells should have more chloride ions outside the cell than inside. So it's not strictly a balance per se, right? But it's achieving the correct sort of ratio of where these ions are located. And the chloride ions will be outside the cell, and they will then attract water out of the cell into the mucus layer. It turns out it's important for the mucus layer to be kept moist because it protects cells by trapping any inhaled particles and bacteria. And if that mucus layer is moist, then the cilia can work really well to sweep away the harmful debris. So in the actual flash thing, you can actually see those cilia, those little lines on the outside of the cell, little yellow lines protruding from the membrane, sweeping back and forth and clearing out the debris. Now, what happens to the function of this CFTR chloride ion channel when there's that phenylalanine deletion? The bottom line is that the chloride ions are going to remain inside the cell instead of outside. So it's the chloride ion transporter, right? So if it can't transport those chloride ions, they stay inside the cell. They can't get outside. And as a result, that means there's too few chloride ions in that mucus layer and too many sodium ions move into the cell. So the movement of the sodium ions into the cell causes water to reverse direction. And the water flows back into the cell, which means that mucus layer dries out, and that's shown by that yellow color of the mucus. When the mucus layer is dried out, it's going to become thick and sticky, and it prevents the cilia from clearing debris. And this allows that debris to accumulate and then cause infections in individuals with cystic fibrosis. Since all of the cells of a cystic fibrosis patient have that defective protein, large quantities of thick, sticky mucus build up throughout the lungs and the other organs. And so this results in the severity of symptoms that are seen in cystic fibrosis patients. Okay, so I wanted to go through that because that really explains what is molecular pathology, and that's what you're going to want to address for section 5 of your paper. Why does a genetic change result in a clinical phenotype? And so parts A through D of section 5 kind of walk you through how you're going to explain the molecular pathology for your disease. Okay, the last thing I want to talk about briefly today is kind of switching gears and now talking about different approaches to clone disease genes for complex traits. So the first thing I want to talk about are these genome-wide association studies. So if you are working on a complex disease, you are probably very likely to come across some GWAS, right? Genome-wide association studies. So these are approaches that because there are multiple genes involved, we can't take the usual approach. So instead, you rapidly scan these markers across the genomes of many people. And you're just looking for genetic variations associated with the disease, not necessarily the disease gene itself right away, but you're just trying to correlate and find genetic variations that are found in people that have the disease. Once you identify these new genetic associations, then researchers can progress to using that information to try to detect and treat and prevent the disease. So in red at the bottom, I just again want to emphasize that these GWAs are particularly useful to find genetic variations that contribute to common complex diseases like asthma, cancer, diabetes, heart disease, and mental illness. Okay, so what are these markers? The most commonly used type of marker are SNPs, single nucleotide polymorphisms. We described these briefly in an earlier PowerPoint. And here is an example of how SNPs were found to be associated with coronary artery disease. So the most common approach is a case control setup, where you're going to compare two large groups of individuals, a healthy control group and one case group that's affected by a disease. And for all of the individuals in each of these groups, you're going to genotype them for the majority of the commonly known SNPs, which is 1 million or more different markers, percent with a higher p-value. So you're looking at millions of those SNPs to try to identify some genetic variants. There is some controversy with GWAs. Some people say that these variants account for only a small fraction of the overall genetic contribution. They're just a small part of increasing risk. For a disease that affects 2% of the population, if you only have one of these variants, it means your risk increases from 2.2 to 3.4%, and they're suggesting or arguing that that's not a very large difference. And some people say that these GWAs are not as good at predicting risk as had been hoped. But other people say there's been actually really substantial contributions, and there's a couple examples listed here. Age-related macular degeneration we now understand that involves the complement pathway, so that's a breakthrough. And for Crohn's disease, we now know that autophagy and the IL-23 pathways are involved. So again, these are things that we wouldn't know to be looking at if we hadn't done these GWA studies. So basically, there are definitely clear success stories that we now have improved treatments and potentially leading to prevention of some of these complex diseases. If we just do a side-by-side comparison and we kind of contrast a monogenic disease on the left with complex diseases on the right, as we think about just what the approaches are for mapping and cloning disease genes, we've already gone through the approach for cystic fibrosis, which is common for monogenic diseases, where number one, you do some mapping. You try to figure out what chromosome and what part of the chromosome the gene is on using linkage studies and lots of pedigrees. And then after mapping, you can use positional cloning strategies, narrow down the region, and then identify DNA fragments that meet lots of the criteria, expected for the gene associated with the disease, and then you can prove or validate you have the correct gene by finding patient-specific mutations when you do DNA sequencing. The second thing is to rescue the normal phenotype in some sort of mutant, and you can use an animal model to show similar symptoms when you knock out the gene. In contrast with complex diseases, the path can be quite different. Sometimes the proteins are identified before the gene is cloned or before the gene is mapped, and hemophilia is a classic example of this. So this is a very kind of flipped order of how the information was obtained. Secondly, if you know the normal function of the gene, you can sometimes create a cell line with a mutation and then try rescuing the phenotype by transforming in random DNA fragments and get the gene that way. So that's kind of also a flipped order of first understanding function and then identifying the gene. And for some diseases, if there are chromosomal mutations, that can be a shortcut. So if you do a karyotype and you're looking at the chromosomes of patients compared to normal and you see differences, then you may be able to quickly map the gene to a part of a particular chromosome. So that kind of shortcut was helpful in cloning the disease genes for progeria and Duchenne muscular dystrophy. And then just a reminder again that those GWA studies have been commonly used in complex diseases to map and identify associated genes that increase risk or somehow promote the disease state. So, regardless of which approach is taken, expression patterns of the candidate genes are useful. So you would expect to see expression in tissues that are affected in patients. For cystic fibrosis, we would expect to see CFTR. That gene should be expressed, meaning we should see RNA and protein in lung tissue, for example. The methods to detect expression, very commonly, RNA is first looked at, and that could be using in situ hybridization or a northern blot, sometimes also called an RNA blot. Or to view protein, you could do antibody staining or a Western blot. Animal models are almost always useful. It's helpful to determine the loss of function phenotype. You can either do RNAi to knock down expression of the gene or do something like a mouse knockout. And this is going to be helpful in determining the function of the gene. And after cloning and confirming the gene, the next steps for all of these diseases are to understand the function of the normal protein encoded by the gene and the molecular pathology. What goes wrong when there is a mutation? Okay, so just to wrap up, you should try to start working on parts 4 and 5 of your paper as soon as you can. I know you're working on the draft of parts 1 through 3, but again, you're going to need to use OMIM to find papers that have primary research articles that have experiments that have led to the mapping, cloning, our understanding of gene function and molecular pathology. So again, interlibrary loan may take a day or two, so please try to identify those papers as soon as you can, and that draft is going to be due Thursday, April 9th. Let me know if you have questions, as always, and have a great day.